All right, today we're diving into one of the most incredible scientific journeys ever undertaken, the Human Genome Project. Seriously, think about it. What if you had the complete instruction manual for a human being? Every single detail. Well, that's exactly what this project set out to discover.
But to really get a handle on the sheer scale of what we're talking about, let's start with a question. If you are going to write down every single biological instruction needed to build and operate, well, just how big would that book be?
The answer is just staggering. The human genome has about 3 billion chemical base pairs. You can think of these as the letters in our genetic alphabet, A, T, C, and G. So that's 3 times billion of these letters, all spelling out everything from your eye color to how you process medicine. To put that number into perspective, let's say you wanted to print it all out. You would need a library of 3,300 books, and each one of those books would be 1,000 pages long. All of that just to hold the genetic sequence of a single person. That's the mountain of data scientists were setting out to map. Now, a project this huge, well, it comes with a pretty massive price tag. The first estimate for the total cost was somewhere around $9 billion. That really shows you the global commitment and the kind of resources it took to even attempt something like this. So you've got to be wondering, right? Why? Why go through all this trouble, spend all this money on such an incredibly complex project? What was the point? You know, the goals of the Human Genome Project went way beyond just reading a long string of letters. Of course, they wanted to identify all our genes and sequence those 3 billion base pairs, but the vision was so much bigger. They wanted to build the tools for a whole new era of biology, and crucially, make all that data public for any researcher anywhere in the world. And I think this is so important. They set aside money from the very beginning to tackle the tough ethical, legal, and social issues, or LC as they called it, that would pop up from having this kind of powerful knowledge. Okay, so we've got the why, but how on earth did they actually do it? I mean, how do you read 3 billion letter code that's packed into something microscopically small? Let's get into the how. This image here gives us a fantastic bird's eye view of the whole process. It all kicks off with a single human cell. From there, scientists zoom way into the chromosomes and then to the DNA itself. After that, it's this whole multi-step journey to turn that physical DNA molecule into digital data a computer can read. Let's break that down a bit. Now, before we get into the nitty-gritty, you should know there were kind of 2 main camps on how to do this. One approach was like a shortcut. Just sequence the parts of the DNA that are actively being used as genes. The other approach, the one the Public International Project chose, was to do the whole shebang, sequence everything, the genes and all the DNA in between them. They wanted to map the entire landscape, not just the cities. All right, so here's the basic workflow. First, you get the DNA out of a cell. Now, you can't just read a 3 billion letter string from end to end, so you have to chop it up into smaller, random, more manageable pieces. Then, and this is a key step, you make millions and millions of copies of each little piece. After that, automated machines read the genetic letters on every single fragment. And finally, you hand it all over to powerful computers that act like the ultimate puzzle solvers, finding where the ends overlap and piecing everything back together in the correct order. So I mentioned vectors for making copies. What are those? Well, think of them like tiny molecular delivery trucks. Scientists would stick a fragment of human DNA into a vector, which would then carry that DNA into a host cell, like a bacterium or even yeast. And then, as that little host cell divides and multiplies, it also copies the human DNA fragment right along with its own. The real workhorses for this were called BACs, which stands for bacterial artificial chromosomes, and YACs, or yeast artificial chromosomes. As you can imagine, sequencing all this DNA created an absolute mountain of data, way too much for humans to ever sort through on their own. And this challenge actually gave birth to a whole new field, bioinformatics. It's basically where biology meets high-speed computer science. And it was the absolutely essential tool for storing, analyzing, and just making sense of the billions of data points the project was churning out. So the project was officially completed back in 2003, a huge technical win. But the real excitement was in what they found. What did reading our own instruction book for the very first time actually teach us about ourselves? Let me tell you, some of the findings were not what anyone expected. So let's get to it. After all that time, all that effort, all that money, what were the big headlines? What did we discover from reading our own genetic code? Here is the first jaw-dropper. Less than 2% of our entire genome actually contains the recipes for making proteins, the building blocks of our bodies. For decades, scientists called the other 98% junk DNA. Well, we now know it is definitely not junk. It's full of complex switches and dials that tell our genes when to turn on and off. Understanding this so-called junk is still one of the biggest areas of research today. This next discovery is, well, it's pretty profound. It turns out that any two human beings on this planet are 99.9% genetically identical. All of the incredible diversity we see in our species, all of it, is packed into that tiny, tiny 0.1% difference. And here's another shocker, the total number of genes we have. Before the project, scientists were guessing we'd have maybe 80,000, maybe even 100,000 genes. The final number? Only around 30,000. That's not a whole lot more than a fruit fly. This discovery totally flipped our understanding of complexity on its head. It showed us it's not just about how many genes you have, it's about how you use them.
So what's in that little 0.1% that makes us different? Well, a lot of it comes down to these things called single nucleotide polymorphisms, or SNPs for short. You can think of them as tiny one-letter typos in the genetic code that vary from person to person. Scientists have found millions of these SNPs, and they're incredibly useful. They act like little signposts that help us trace human migration patterns and, really importantly, help us pinpoint the genetic roots of many diseases.
This table gives us a really nice snapshot of our genome stats. A couple of things really jump out at me here. Look at the huge range in gene size. The average is about 3,000 letters, but then you have the monster dystrophin gene at 2.4 times million letters long. And it also shows that our genes aren't spread out evenly. Chromosome 1 is absolutely packed with them, while the Y chromosome has very few in comparison. Okay, so we've got this sequence and the surprising discoveries, but what has been the project's real-world legacy? What can we actually do now that we couldn't do before? Let's get to the big so what. Honestly, the impact has been nothing short of revolutionary. Before the Human Genome Project, scientists studied genes mostly one by one. Now, with the full genome in hand, we can look at the entire system all at once. This has completely changed how we diagnose and understand diseases. It's given us incredibly powerful tools for forensic science, and it has allowed us to map our ancient human history with stunning accuracy. There's a quote from the original project documents that I think just nails it. They said the project would enable a radically new approach to biological research. It was never just about getting a sequence. The whole point was to create a foundational tool. And it has absolutely delivered on that promise. And then some. For the first time in the history of life on Earth, a species has read its own instruction book. The scientific quest to get here was incredible, but the next chapter, well, that's ours to write. The question for us is no longer, what does our code say? The question is, what will we choose to do with this knowledge? How will we shape our future?